So we'll get started. Hello, everyone. My name is Stephanie Hagstrom. I'm the Director of Community Development for the Research Data Alliance US. Today's webinar is part of our 2021 webinar series hosted by RDA US, and the webinar being presented is in the series. This series is a joint effort of the RDA Physical Samples Interest Group and ESIP Physical Sample Cluster Group. We're really excited about this joint venture, and this is, Esther, is this our third or second? Second? Can't remember, but anyway, so I want to give you a little bit of background about RDA and RDA US, and then I'll turn it over to Megan for just a quick overview of ESIP, and then we'll get going. So the Research Data Alliance is a community of over 11,970 data experts from 145 countries around the globe, and they collaborate together to build social and technical bridges to enable open sharing and the reuse of data. Members of the RDA come together through self-formed focus working groups and exploratory interest groups to exchange knowledge, share discoveries. They discuss barriers, potential solutions, define policies, test standards, and many of the 97 current active groups have produced outputs to enhance and facilitate global data sharing and reuse. The RDA also has 26 plus national groups that support initiatives of the members of those regions and provide a platform for those regions to interact on a global level. There are 4 regional groups of the RDA that provide resources to support the RDA global operations. The RDA US is where I work, is one of those recognized regions and is represented by 2,500 members who help strengthen the data science infrastructure community. We are very excited to have a partnering arrangement with ESIP on this project, and I'm going to turn it over to Megan really quickly for her to give a little background on ESIP.
Thank you, Stephanie. So as she mentioned, I'm Megan Carter. I'm ESIP's Community Director, and we too are excited for this webinar series, this joint collaboration to continue. The Earth Science Information Partners, or ESIP, is a global community of earth science data professionals, and they work to advance the usefulness and impact of data to address pressing environmental challenges on a global scale and to fuel new discoveries. So for more than 20 years, ESIP has helped members of the earth science data community to find each other across traditional boundaries, both to leverage each other's expertise and to work together on common data challenges and opportunities. ESIP fosters rich collaborative experiences, which include 2 annual conferences. We actually just concluded one in July. The ESIP Lab, which supports the development of innovative applied earth science technologies, and 30+ community-driven virtual collaboration areas. And these span a very wide range of topics, from more technical topics like machine learning and cloud computing, to application areas like agriculture and climate and disasters, to physical sample curation, you name it, we probably have a group for it. Anyone is welcome to join, and I'm happy to speak to any of you all if you're interested in participating.
Great, thanks, Megan. Okay, so I have a little bit of housekeeping and then we'll get started. So first of all, we will be recording this presentation. We are recording this presentation. And we will be uploading this recording to the website afterwards on the RDA website, somewhere around here. So go to this webinar's website and it'll be there. We'll also upload all the slides to the same place. We have allocated some time at the end of the webinar for questions. There are 3 ways that you can interact and ask questions of the presenter. One is to, in your Zoom place, I don't know exactly where, there's a place where you can raise your hand. If you raise your hand, we can unmute you and you can ask the question directly to the presenter. If you prefer, you can put a question into the Q&A area, which is at the bottom of your Zoom screen, or you can post questions to your chat, and we'll pick those up as well. If for some reason we run out of time at the end of the presentation, or the Q&A, excuse me, then we will take those questions and put them on a Excel spreadsheet or a Google spreadsheet, and we'll get the answers to those and send them to all of you. Okay, so I will turn this over just wanted to say that today's webinar is titled RRIDs, a way to track samples through the scientific literature. And I want to thank all of you for taking time out of your schedule. And I'm going to turn it over to Esther now to introduce our presenter today.
Thanks, Stephanie. And thanks also to the RDA US for hosting this second webinar in the webinar series on physical samples. So as mentioned, the series is jointly presented by two groups specifically within the RDA and ESIP focused on physical samples. So the ESIP physical sample curation cluster was initiated in 2020, and it aims to enhance the discoverability, access, and use of physical sample collections. And the RDA Physical Samples and Collection Group was initiated in 2017 during the 10th RDA plenary already. The group aims to facilitate cross-domain exchange on key issues and recommended practices related to the digital representation of physical samples and collections. In the past month, for example, group members have worked on a physical sample version of the RDA 23 Things output, with a deadline for group contributions on the 24th of August, after which the output will, of course, like normally with RDA outputs, be up for community review. So since these two groups have a very similar focus, we're now co-organizing this webinar series to increase the awareness of the importance of physical samples in research. And during the first webinar on electronic lab notebooks on the 30th of June, the topic of persistent identifiers for physical samples was raised multiple times. So you'll be excited to see that for today's webinar, we'll be focusing on that topic. And so from the earth sciences, there has been a focus in the past years on the use of the International Geosample Number, or the IGSN, for the use of persistent identifiers for physical samples. And there's currently some work on the way to make this persistent identifier more applicable to other disciplines as well. So today we'll be focusing on another discipline, which has a biomedical focus. And we're going to hear from Anita how this field has successfully implemented persistent identifiers for resources. And what everyone else can learn from these challenges and lessons learned. So I'm going to ask Anita if you can turn on your camera.
Yeah, absolutely.
And already share your screen. And then I'm just going to briefly introduce you. So it's my pleasure to introduce today's speaker, Anita Bondowski. Anita is one of the leads of the FAIR Data Informatics Lab at the University of California, San Diego, where she is based at the Department of Neuroscience. She is also the co-founder and CEO of SciCrunch Inc., which is an initiative that provides platforms and tools to track research regions and improve transparency and reproducibility in scientific research. So we are very excited to hear from her experience and apply these lessons learned into our own scientific domains. And while Anita is speaking, please do add your questions in the chat or the Q&A section. And as a reminder, we can unmute you to ask your question, but please wait until the end of the presentation for this. I would now like to hand over the digital floor to Anita. Please take it away.
Thank you so much. I appreciate the nice introduction, Esther, and hi, Stephanie. So we're going to be talking about research resource identifiers and the scientific literature. So really the impetus for a lot of this came from the National Institutes of Health. And I'm sorry, I didn't define that on my slide. For those of you in the physical sciences, NIH just means National Institutes of Health, and that is who we ask for money across biomedicine. So what they said to us in about 2008 is they said something inside of a contract that we had at the time in the lab, that people are wasting a lot of time looking for antibodies. Antibody is a type of reagent. I'll talk a lot more about that. Why can't you help them by creating an algorithm to actually find those? And so we tried. And the first thing that we did is we actually tried without creating an algorithm first, because we needed to see what the literature actually looked like. And this is what the literature looks like. So this is actually a much more recent paper than one of the ones we looked at. But what it's showing is that in the method section, where the authors of this paper are actually talking about confocal laser scanning microscopy, It's a technique that's often used with antibodies. There's a description of an antibody here, and this is one of the reagents, and it's a mouse anti-flag. So this antibody was raised in mouse against a protein called flag, and it was purchased from Sigma. This is Sigma Aldrich. Here's its dilution, and that is what we have for this. So if you go to Sigma, and this is now called millipore Sigma, what you find is that there are 17 anti-flag mouse antibodies. And so if there's no identifier of any sort, then this reagent is actually not findable. And that makes this paper not reproducible for a really dumb reason. Okay, so just to give you a really quick overview, just in case you're not a biological scientist, about what an antibody is. So here's our friend, the rabbit. And the rabbit has been introduced to a particular protein of interest or a disease of interest or something else. And then that rabbit produces an immune response. That immune response is partially mediated by a cell type called a B cell. So what you can do is you can extract blood out of this animal or human or whoever you're actually immunizing. And you can extract some of these B cells. Now, each B cell only produces one type of antibody. And the way that it does it is it recognizes a particular epitope of that protein that was introduced. And then it starts to spew out a lot of these antibodies into the bloodstream. Those antibodies mount an immune response to this rabbit, to this protein in this particular animal. But what these B cells can also do is they can actually be fused with a cancer cell. So this B cell can be fused with this cancer cell, and it actually forms a new hybridoma. It's called a hybridoma. And that hybridoma has the function of being completely immortal. So now what can you do with an immortal cell that also produces this lovely reagent? Well, you can plate it, you can use it for experiments, you can freeze it, you can thaw it. It will still continue to produce the exact same reagent here, this antibody. And these antibodies are a way for us to treat COVID-19. It's one of the treatments that was received by multiple people now. It's a way to recognize the spike protein, but it's also a way to find any target protein that you might be looking for in your sample. And of course, you can buy them from many, many different companies under a particular catalog number. Okay, so the question that we had initially was which antibodies are being used in a particular paper though? Because unfortunately, unlike the lovely representation here, we actually have antibodies that don't work very well. And I think that was demonstrated early on in the COVID epidemic, where we had some antibody tests that failed. They failed specificity and they failed other tests. So that's unfortunately a very common thing with antibodies. For the research use only antibodies, it has been estimated that about half of them fail. And there are various reasons for that. I won't go into that very deeply. But for the ones that work, they tend to be the ones that are being published in the scientific literature. But as you can see, the way that many people describe these antibodies, there are multiple antibodies that could have been used instead of just the one that was actually used by the authors. So which reagents are actually being used in the paper? It's a very important question that we couldn't really answer. And then which other papers actually are using this antibody? These are two really super simple, trivial questions that we could not answer in 2008. So we didn't even know how many antibodies are out there for people to use. So we ended up having to build the antibody registry, which currently covers about 95% of the antibodies that are used in the literature. And I'll show you how we know that. We gathered data initially in 2010 from about 250 different commercial vendors and also non-commercial vendors. We currently work with about 30 of those vendors to regularly update data. So companies like Thermo Fisher send us a list of their 100,000 antibodies. We update that. We give back the RRIDs to them, the identifiers to them, and we do that every quarter. We also work with about 3,000 labs to register their antibodies. we give all of these antibodies an RRID, a specific unique identifier. And by the way, in order to get all of this to actually work, our turnaround tends to be about 1 business day. Okay, so if we just have a catalog number, why isn't that good enough, right? So just asking people to use a catalog number from a company, big company like Sigma or Thermo or one of these, why isn't it good enough to actually just say, hey, this is what I used? Well, let me demonstrate that to you by looking just for this one antibody. And this is this one RRID. You can see that the number here I'm searching for is AB 2017-8887. This antibody is against the protein NF-kappa B. And this is currently being sold through Millipore, and this is actually Millipore Sigma. But the provenance of this antibody is that it was actually created in an academic lab. And that academic lab licensed this product out to a company called Boehringer. Boehringer gave it a catalog number 1697838. There's a space in there. And then Boehringer, after a couple of years, was bought by Roche. And Roche said, no, we don't like spaces in our catalog. So our catalog number has now become 1697838, no space. Okay, that kind of looks the same, but Roche, after a couple of years, decided that the antibody field was not part of their business model. So what they did is they sold off all of their antibody reagents to a company called Chemicon. And Chemicon was, in a couple of years, bought by a company called Millipore. Now, that actually was sort of a hostile takeover. And so somebody had done something with these lovely cell lines, and they didn't work very well anymore. And so Millipore had to go back and re-extract out from a deep freeze these cell lines that I told you about before that could generate these new antibodies. So what happened is the original creator of this antibody had actually had to publish a paper and say, hey, this antibody isn't quite working the way it was supposed to. It's not working the way that it was working before. And Millipore knew that. They figured it out, and now this antibody is okay again. But there was a period of time when this was happening. Now, mergers and acquisitions don't actually stop there. So this antibody went from Chemicon to Millipore, then to a company called EMB. And now it was that company, Aggregate, had been bought by Sigma, but in fact, this is still marketed under the Millipore name. So the problem is that when Chemicon actually bought this antibody, it changed the number of the catalog number from this numerical string to MAB 3026. Thankfully, Sigma continues to use the same number, but there's no telling what will happen in the future. So what you can see is that there's a really compelling reason to actually use a persistent unique identifier when we know the provenance of a particular reagent. And if you want to say, oh, that should be easy if there's only a handful of these, but in fact, there's about 2.5 million. All right, so this is the full citation of what we're asking people to do. So we're saying, okay, this is a Millipore product, which is currently in the Sigma umbrella. There's its catalog number, and there's the persistent unique identifier, which they have to look up by going to one place, one portal. So the way that this whole thing looks is the company name, catalog number, and of course the identifier from an authority, from the authority that is recognized for that type of research resource. So for the IGSN community, this identifier from authority would be the IGSN number if the IGSN numbers were ever part of the RRID initiative. Okay, so what we have done is we actually gathered a lot of these authorities. So one of the authorities here is the antibody registry that I just told you about, but there are many other authorities. We don't control them. Cellosaurus is at the Swiss Institute of Bioinformatics, and it's being run out of Europe. Many of these mouse repositories are either in the United States, some of them are in Japan, some of them are in Europe. But there's a whole lot of these different kinds of critters, different bits of DNA, different kinds of reagents that are for sale around the world. And those bits of reagents each have a set of metadata that's associated with each one of those identifiers. And that metadata is all pulled together and it's searchable in one single portal. The other information in that same portal with those same records actually comes from either the literature itself, and I'll show you how some of that happens in a few slides, where we have a full pipeline that actually grabs the information that a particular paper is citing a particular reagent. We also have a bunch of projects that talk about whether or not one of these reagents works well or doesn't work well. And I'll show you some of those things again in a couple of slides. So how did the RRID project actually launch? So in 2012, we actually had a first meeting at some place called the Commander's Palace, which is exactly what it sounds like, a really fancy food place, at the Society for Neuroscience, our big scientific meeting. And what we learned in that first meeting is that the way to an editor's heart is through their stomach. So we actually paid for a very fancy dinner for the editors-in-chief of 25 of the biggest journals in neuroscience. And we got them to show up, which was really great. The second meeting was about 6 months later, and it was sponsored by our friends and colleagues at the NIDA, the National Institutes of Drug Abuse. And that meeting was a 2-day workshop at the National Institutes of Health, where we again had these 25 journal editors-in-chief in the same room arguing with us and with each other about how such an RRID initiative would never ever work. So later that year in November, at the next Society for Neuroscience meeting, we actually got a start time for a 3-month pilot project that was going to start with those 25 journals, and the journals would actually ask authors to provide RRIDs. We were going to gather the data up and figure out if this had worked. So in about 2 months over the holidays, we actually needed to aggregate all the information for all of the antibodies, the tools that were going to be part of this initiative, 25 different kinds of organisms. We needed to put up a website that authors could actually understand, that was very easy to use. And of course, we had to put in place a help desk because people would inevitably get confused. By the way, there was no additional funding for us to do any of this. It was kind of nice that only 2 journals started on time because we weren't completely ready within 2 months, but they did start on time and we were ready enough, I suppose. The good news is that we are currently in the 7th year of this 3-month pilot project, and our latest journals that have joined are Nature, Science, Cell, and eLife, which are all on board. Okay, so what are the journals actually asking authors to do? So basically the journal has some instructions to authors. Sometimes they have a letter, sometimes they have a part of a checklist, which directs the author to the RRID portal. So this is an example of the RRID portal with an executed search for this particular core facility, but they are going to actually be picking up, the author is going to be picking up all of their resources, including things like antibodies, cell lines, software tools, and other things. And they're going to search for those, and they're going to copy this bit of text from the Cite This button. It's also here. And they are going to paste that into their manuscript and move along their merry business and publish the paper. Now that paper becomes data because it becomes searchable by that identifier. And the kinds of things we've included are antibodies. There's 2.5 million of those. Organism stocks, there's now close to 600,000 of these animals that are available to purchase. There are over 100,000 cell lines, and there are nearly 100,000 plasmids, which are bits of DNA. And there are various statistical tools, core facilities, etc. Woo, not sure what happened there. All right, here we go. So RRIDs are currently part of the journal article tagging suite. That's the tagging library that's used by journal publishers to structure their content. And that is new for last year, actually. This became part of that particular release. So here's the resource wrap, and this mouse has been forever enshrined in the in the documentation for how to structure an article into XML. Science Magazine also recently began to require that a material design analysis reporting checklist, also called the MDAR reproducibility checklist, be filled out by every single paper that is accepted into science. The first thing that this checklist asks for are RRIDs for antibodies, cell lines, and other organisms. So this is how we're actually getting compliance from authors from Science Magazine. The RRIDs are also present in the catalog. So this is a catalog for antibodies from a commercial vendor. This is a catalog entry for a mouse from the UC Davis Stock Center. And here is the Cellosaurus page for a particular cell line. The NIH continues to do some things to help authors also find RRIDs. Here's the resource sharing plan, where the grantees are actually being directed to the RRID portal to register all their new research reagents. So if they create an antibody, if they create an organism, then they are directed to basically register that with the appropriate authority. We also really like robots. So we were asked to build robots initially, and we're still building them. So what we actually have done is we've actually looked at all of the papers that have an RRID, which is incredibly easy because all you have to do is go to Google Scholar and type in RRID, and you get back about 30,000 results. Just about all of them are good for who's actually using what kind of reagents. And so there's a robot that we have called Cybot, which looks for things in the text that look like RRIDs. It takes this bit of information here, this number, and submits that to a database, essentially through an API call. And it pulls back that information in this annotation layer, which then says, hey, here is the actual information that's associated with this, with this particular ID. And then the curator can look at that and say, oh, cell signaling technology number 3450, cool, that's exactly what the database says as well. That matches, good to go. And basically gives that annotation a thumbs up. And then another robot actually takes that information and picks it up and tweets about it, which is very cool because then that tweet becomes part of the altmetric record for this particular paper, which is a really nice way to say thank you to this set of authors who have included and done this extra work to include these identifiers in their papers. So what we're doing with this new RRID robot is that we're actually tweeting messages of very positive joy that they have done a nice thing for science to improve reproducibility. So this is kind of a virtuous cycle. And if you want to go to Twitter and follow this robot, you would be joining, a good handful of your friends. So in order to, but in order to actually see these tweets, you can actually just see the paper altmetric page, which is where a lot of our views are actually coming from. So, while only about, I think, 45 or so people actually follow the RRID robot, these tweets have been viewed several 100,000 times because they come out really quickly after the paper is released. So while people are still looking at this paper and looking at the tweets that people are sending about it, we can actually get this turned around. So after we find a paper, it can take, less than 1 day to actually do this annotation and release the tweet using our lovely robots there. So we still we enter into the kind of conversation on Twitter about this paper. Okay, so the question is, did this work? And the answer is, well, over 1,000 journals actually have RRID containing papers. Some of the journals actually do this nice linking to RRIDs. So here's the STAR methods table, which was created, which asks specifically for RRIDs for each of the different types of reagents that they're for. And they do typeset that, so they actually create the links for us. The same thing with eLife. Journal of Neuroscience, our favorite society journal, does talk about RRIDs in their, and they actually send letters. Endocrinology Society actually requires RRIDs for antibodies, at least, and others do as well. Okay, so this is what one of these RRIDs looks like. So here's a mouse, and this mouse is linked. That link is put in by the journal staff itself. It doesn't have to go in by the author. And if you click on that, we actually direct you to the Jackson Laboratory material data sheet for this particular animal. So then this paper becomes much more reproducible, just in the sense that you can actually really easily figure out exactly what these authors are using. Okay, let's just quickly look at the growth over time. So in 2014, we had about 250 papers altogether. In 2015, that nearly doubled, although you can't really see that very well. And then that went up really fast between 2016 and 2017, and it continues to go up. The top journal right now for the largest number of individual RRIDs that have been noted is Cell Reports. This is one of these like high volume publishers at Cell Press, and there are 54,000 RRIDs that have been attributed to different articles at Cell Reports, but of course eLife is a close second here with 38,000. And then there's a whole lot of other journals, including the Journal of Neuroscience and this tiny journal, the Journal of Comparative Neurology, which doesn't publish very many papers at all, but they really insist very strongly that all of them have RRIDs, so they're in there too. Okay, so using identifiers for resources actually does make better, more reproducible papers. And the reason that we say that is because with the first 100 papers we looked at, which this figure is from, we showed very clearly that in the same journals before the RRID initiative and after the RRID initiative, we get as basically an increase in reagent identifiability of about 40%. So it starts at, a human being looking for these antibodies can find them half the time in the same journal versus they can find them about 90% of the time with RRIDs. So that's really great. But let me show you where this becomes really critical. So cell lines can be misdiagnosed. And how weird is that? But what a cell line is, it is a reagent, but it's also a part of a human being often. Sometimes it's part of a mouse, but very often it's part of a human being. And those human beings can be misdiagnosed. I'm sure that all of you have probably some example of this in your friends and family. Someone was misdiagnosed with a particular disease. Often we take out a tumor, we plate that, we can actually use this. Now you can buy a whole lot of these cell lines, but that human being is also associated with a set of health records where people write down what kind of information that they know about both this patient and the tumor. Now, if you're going to be using some of these tumor cells to discover, what treatment is effective against cancer A, what you want to do is you want to find the cell lines that are supposedly about cancer A, and then you want to put a bunch of treatments on them and figure out where they die most. And if you do that, that's great. And then you get your paper and all is well. But what if that cancer A is actually cancer B? So what happens if you've got the wrong cancer is that there is a significant problem, a muddling of the waters in the scientific literature, because now you have discovered that, in fact, you say that cancer A is being affected by these drugs, but in fact, you're using a cell line that is about cancer B, that expresses cancer B. And there's plenty of examples of that. And when we've looked across the scientific literature across 150,000 papers that use cell lines, we actually found that about 16% of the papers have a problem with something like a misdiagnosis or another kind of problem that was identified by the International Cell Line Authentication Committee, which is an international group of scientists that really deal with cancer cell lines and which ones are actually, having some kind of a problem. So this is an example of one of those cell lines, and what you see here, and some of these are in red now, that the comment actually says this is a problematic cell line. This one's been contaminated. It's actually been shown to be a derivative of the HeLa cell by using sequencing. And so when using this cell line, this is not an adenocarcinoma. This is actually a HeLa cell. So this cell line should not be used. Other misidentified cell lines can be used, but they can't be used for cancer A. They must be used for cancer B, so that we understand what we're actually looking at. So what happens with RRIDs, right, is that at least in these 634 papers, these were the first 634 that we had published, we found that instead of 16% of the literature that was using one of these misidentified or otherwise problematic cell lines, we only had 5%. And when we looked deeply into these 5%, we only found one cell line that was used that was truly problematic. The rest of them were actually just misidentified and okay to use. So RRIDs, that second step, where right before you publish, you have to look up an extra number, which I'm sure is really problematic. It's a pain in the butt for everybody. But if you do that, if you ask people to do that step, they can see these warnings and it cleans up the literature. So this is a huge boon to cleaning up the cell line literature, which is why a lot of cancer journals have now joined the RRID initiative. So going back to antibody reagents, again, we're looking here at about 300,000 antibodies from 682 different journals. This was published last year by my student, Joe. And basically we're looking at, can this antibody be identified? And we explored that by looking at the literature in the PubMed Central database. And we found every antibody from every paper in every year that we could. And we text mined those out. And we found a catalog number essentially associated with about 10 to 15% of the literature in the 90s. And that went up to about 20%, 25% here in 2014 when the RRID project started. And then what you can see is that over the next few years, there was maybe a bit of a shift, but not much. But as soon as 2016 started, we actually have this fairly large shift, this improvement in the inclusion of catalog numbers in the scientific literature overall. So you can see that this shift is actually fairly fast in the last couple of years. And where this comes from is this implementation of the RRID initiative. So basically you've got some of these big journals like eLife and Cell actually implementing RRIDs, and it drives very quickly the identifiability, which was about average before the initiative, to 90+%. And that actually is driving increased identifiability in the entire literature, and probably because of spread of, basically somebody puts a paper into Cell, gets rejected, goes into PLOS One. Okay, great. Now that gets published, so even PLOS One and OncoTarget or whoever else is actually getting, an increase in the, ability for somebody to be able to find those reagents. Okay, so this is great. Why is the reagent problem not yet solved? Well, and why does PLOS One not actually enforce RRIDs, where they have actually changed their instructions to authors, but we actually know, based on this graph, nobody reads those. So what we did is we said, okay, it's too hard to do. The journal editors, especially at PLOS One, really don't have any time to be enforcing these kinds of reporting criteria. Whereas, Science and Nature maybe can do that, but it's really hard for these smaller journals or the high volume journals to be able to do that. So what we did is we created a tool called Cyscor. And this is a commercial tool. So this is my conflict of interest hat. But this tool is accessible if you log into the tool using your ORCID ID, it's free to use. And what we see is that we do have partners, American Physiological Society, the American Association for Cancer Research, Microbiology Society, and Carter are all partners using this tool in some way or another. It does a lot of things in addition to checking for RRIDs, but it certainly does check for RRIDs. It checks for statements in the paper that are supposed to be using RRIDs, but don't use RRIDs necessarily, and it tries to suggest them. So the other thing that it does is it also gives the author a score, which is kind of fun, because it's just a methods completeness score. Essentially, it's how many of these rigor criteria, how many RRIDs are there versus how many are there supposed to be. And so what it does is it takes the methods section and it produces things like the materials design analysis reporting checklist. And it does that within a minute or so. Okay. Now, because we have this tool, we can actually do some really cool things with it. So we looked at the journal Nature, and this is the journal Nature. This is essentially the percentage of reagents, for example, that are being found in that journal. And so what we have is we've actually figured out that in 2009, the number of antibodies, see this in orange, that are being found here with catalog numbers is about 10 to 15%. So that's not terribly surprising. It's about average. And then after the implementation of this Nature checklist, which happened in 2013 to about 2015, that percentage of catalog number containing antibody references more than doubles. So it goes up to about 45% by 2019. And we can also look at many other things. We can look at whether or not authors are reporting sex of animals or of patients. And that again goes up from 20% to about 50%. We can see how many experiments are reporting randomization properly and blinding of investigators or subjects. All of these kind of features go up. And then, of course, the average score for presence of all these different criteria also goes up over that time. It doubles from about 2.5 to about 4.5 average points out of 10. So, and if you look at the literature altogether, now here we looked at 1.5, 1.6 nearly million papers. And we're looking at things like the sex of subjects, which really does go up over time, and blinding, and of course, the reagents are getting better, but we certainly have a long ways to go. And by the way, oh, thank you. All right, so I'll go a little bit faster through this because this is not so important. So we created this metric to see if it would be correlated with the impact factor. And the answer is, while that trend line suggests that it's negatively correlated, there's actually no relationship statistically between the impact factor and this metric of what we believe to be quality of methods. So now we have one tool that can traverse the literature and find important things and bug people. And so what we ended up wanting to do is actually bring on a lot of other people. And this is a good chunk of the world's AI experts who meet sometimes and have a bunch of cool tools. And so Cyscor is one tool that I told you about, but there's a whole lot of other tools out there that can point out different bits and pieces of information to authors. And these bits and pieces of information about things like non-colorblind safe graphs and clinical trial IDs and self-acknowledged limitation sentences and other fun things are actually, Cite AI is an important one. It checks for, has the paper actually used a retracted paper in its reference list, and have not acknowledged that. So there's many more to follow, but this was a really cool thing that we were able to do. And we came up with a way to basically look at the COVID preprints that are being put out there. We've put in a lot of information together, and we process all of these preprints now with all of the 9 tools, and we were able to actually publish this work, which was great. So we're letting people know through Twitter how their scores look, and so this is a nice example of that. And again, I think that I'm gonna wrap it up here by just saying thank you very much. I think we've learned a lot about how this, how to put in persistent identifiers into papers and what are the sorts of things that that can actually help with. Okay, so I think, no, I'm not over time. Okay, great. But I think that would be fine.
Thank you so much for this wonderful presentation, Anita. You were right in time. I shouldn't have warned you. Thanks so much. It's really impressive to see how things have developed, and so quickly as well. We already have a couple of questions, but before I go to them, just a brief reminder that you can post your questions in the Q&A or in the chat, or if you would like to raise your hand and ask the question yourself, you can also do that. So please do so. Frank had the first question. And he was wondering whether there are any agencies in Europe or the UK that are advocating or promoting the RRIDs or supporting the RRID initiative.
So I think in terms of, so some of the, the core centers, the stock centers and what have you, that are creating the RRIDs. I pointed out that the cell line repository is actually from Switzerland. It's being taken care of by the former head of Unipro. And that is housed at the Swiss Institute of Bioinformatics. So there's certainly EU support for that. There is EU support for the COMP project, which donates a lot of mice to the to the stock centers, and those are again taken care of by several repositories both in the US and in Europe. But as far as RRIDs, I mean, they're certainly being pushed much more strongly inside of the US than they are inside of the EU, but I welcome, highly welcome any kind of an initiative from the EU governments to help us push and promote RRIDs. So Frank, if you want to contact me, I'm happy to chat.
Wonderful, thanks. And then we had another question from Andrew, who is highlighting the biological collections community. So this community is trying to do something similar for citing specimens in papers and linking these to specimen records in aggregators to support the extended specimen idea. And as with the biomedical community, getting journals and editorial boards on board to support this by requiring links in the articles, etc., is a limiting factor. And he was wondering whether you have any tips and tricks to engage the editors and editorial boards to get on board, besides inviting them to dinner. Besides wining and dining them was the exact phrasing.
Well, wining and dining seemed to be fairly effective, but it was only effective to get a very small pilot on board. But it was, it's definitely, continuing to follow up and really getting some of the big funders on board, because that really helped us, I think, pushing this. We had a 2-day workshop that was paid for by the National Institutes of Health. And what happened there was that, we were actually able to figure out, what we could do, how we can do it, and we could take into, into consideration all of the kinds of things that the journals had issues with, right? So we had all of those kinds of questions like, well, what would happen if, and what happens when, and all of that. So we really had to, in order to get this done, we really, on the informatics side, we really had to have our act together, because, now that there are about 360,000 RRIDs in the public space as a public data. What that means is that there were 360,000 lookups by different authors. And that means that our systems had to be sufficiently robust and reasonably understandable so that individual authors could actually do this task, right? And then the second thing is we had to have that help desk involved. And of course, bringing on the journals now is very easy, because we have all of our act together. We know exactly what to put into the instructions to authors. We know exactly every question that they're going to bring up. And it's all in a slide deck, in a single slide deck, where, because we've been asked the same question over and over again by different journals. I'm happy to share that slide deck with you. But you kind of have to have your act together first. And then you have to make sure that you're going to be standing behind that. So we have the antibody registry and we have another registry, but a lot of these other registries and partners also had to agree to kind of take questions, take animals, take stocks from all the different authors that might be out there. And our turnaround time can't be 2 months, right? We have to respond back to that author very, very quickly. So, I was doing the original curation with the antibody registry, and I noticed, for example, an author would come in, they would register, and their entries were horrible. But, we got those in shape, and, we made them really pretty. We did all the stuff that we needed to do in order to make sure that that entry was good and it workable. And then we would send back an email to the author, and the author would say nothing, right? But then like 6 months later, or a year later, I would send back another email from a really nice entry. And it turned out, oh, wow, this was the same author that had made the really bad entry before. And so even if they don't respond, they appreciate what we did, right? So, and then they came back and they actually made a really nice entry. They paid more attention 6 months later or a year later, and that happens all the time. So we have to be really professional, and we have to be really fast in our response. So little things that I would say are really important. Okay.
I'm not sure those are little things, but yeah, I do hope that they provide some place to start for Andrew or anyone else that would like to start a similar initiative. Did we have an unmuted participant? Because I heard some background noise.
Look, I don't see any hands.
Okay.
We seem to have a few more questions.
Yes, we do, but I just wanted to make space for if anyone wanted to raise their hand and speak. So anyway, I hope that addressed the question from Andrew. We also have a question from Vaida, who is asking, apart from authors or journals that are including RRIDs, what other ways can authors or electronic lab notes journals improve the use of RRIDs? So what can we do?
So ELNs are wonderful tools, and if they can actually, so what I showed was that the we do have our electronic act in hand, in order. So there are lookups that we can do, and we can provide APIs to electronic lab notebook vendors that they could use to mark antibodies or cell lines or what have you that people have in their labs. If they can provide that set of research resources as outputs for a journal article with the RRIDs, as we ask people to cite them, I think that would be wonderful to be able to provide. And that would be, I think, really amazing in general, that to be able to work with the ELN providers to make this step, which unfortunately always happens at 11:30 at night on a weekend, of submitting to the journal and then having to look up all these numbers. So much better if we can get that done beforehand.
Yes, indeed. And perhaps we should also, as authors, improve our practices and not try to submit any articles in the middle of the night. I hope that addressed the question by Vida. We also have an anonymous attendee who is asking whether you've worked with privacy or medical data expert in terms of protecting personally identifiable data.
So personally identifiable data is always really, really difficult to deal with, especially for groups like ours. What we do is we try to get all of the data de-identified before we ever touch it, so that all of our data can be open and public and we don't have to deal with these personally identifiable issues. Of course, with cell lines, they're not actually protected. And that has sparked a lot of problems with some of the community. I don't know if any of you have seen the Oprah's movie about the HeLa cell, which is the immortal life of Henrietta Lacks. It's also a great book, but Oprah made a movie out of it. I highly recommend watching that because it raises a lot of these kind of issues. And a lot of scientists don't really realize when they're working with these cells in a dish, that those cells in a dish had a sex associated with them. They have a gender, they have a family, they have, surviving members of that family. Maybe the patient is gone, long gone in some cases, maybe the patient's still alive. Those bits of a human being are still part of that human being. And so doing things like, fully sequencing and completely opening the HeLa genome is great for science, but it does raise some very contentious questions about the privacy of this family. And so I think we're not going to solve all of that by using identifiers, but there's certainly a lot of legal and ethical issues that we all need to be concerned about.
Thanks for raising that very important issue. And thanks also for the extensive answer, Anita. I think those were all the questions. If we haven't answered your question, I think we can you can get in touch with Anita or send us an email so that we can get back to you. And perhaps you can share your email address in the chat, Anita, for that. And then for now, I would like to thank you very much. And then I would like to hand it over back to Stephanie, who is going to conclude with some closing remarks. Thanks.
Great. Thank you so much. Oh, Anita, that was wonderful. Thank you so much. Full disclosure, I was at FDI Labs for a number of years with Anita, and so it was fun to watch this presentation for me. I want to thank you very much. And Megan Underwood for helping us technically, and for the audience for all your wonderful questions. In closing, I want to bring up that we do have upcoming webinars hosted by RDA US, so keep an eye out for that. The next one up for the highlights for RDA outputs is the Fair Island Project on August 26th. And if you're not already a member of RDA, please join. We would love that. And then lastly, our 18th Research Data Alliance virtual plenary meeting will be in November 3rd through the 18th. And I want to thank, on behalf of RDA and RDA US, thank you all for joining us today. We hope to see you soon. Best wishes to you all. Bye-bye.